thank you everybody for joining us today. Welcome to this edition of the Virtual Pub on Correlated Imaging Series. I wanted to start by introducing myself because I think that many of you won't know me. I'm new to Euro-BioImaging. I started in past November, and I'm working mainly in the AI for Life project, so you will hear a bit more about it in a minute. So our speaker today is Perrine Paul-Gilloteaux. Apologies for my French. Perrine is a computer vision specialist, and she's also an expert in bioimage analysis. She works in France at CNRS as a research engineer. You probably know her, because she's a very active member in the imaging community, and she engaged in plenty of initiatives like COMULIS and UBS as well. Today, she's going to talk about image analysis tools and applications for correlative microscopy. So if you have any questions while she's speaking, please write them in the chat, and we will also have some time at the end for questions that you will have the chance to ask yourself.
So thanks a lot for the invitation, then. And indeed, the purpose of today is to speak about correlative microscopy. And I mean, you're here and you know we are all coming from different fields, and always a common denominator of imaging. And there is plenty of different imaging types. Everyone will actually offer a lot of different information from the structural point of view, dynamics, physiological, functional, chemical information, and this at very different scales or penetration depth also. And then come the natural need indeed to fuse them, or to try to merge these different information and to get the best of all these modalities. And that's the need then to image the exact same specimen, or in clinical, maybe the same patient or the same animal in preclinical, with two or more even complementary modalities in order to have a multi-scale view of the sample, to have different and complementary information, and also because sometimes it's useful to identify a specific rare events or rare region of interest. So there is, I would say, two big solutions for that. So the first one is to use some of this hybrid imaging system, basically system integrating several modalities in one system. So there is plenty of example in the medical field where you can find like a PET scan, a PET MRI, or different system integrating the modality, so you can get from the same system two different informations. That's also coming a lot now in the microscopy field. So at a different scale, just two examples among plenty of them. So, for example, here some example merging confocal fluorescence and photoacoustic on the same system, or here proposing fluorescence microscopy and electron microscopy on the same system as well. The good thing is that it's ready to use. You don't move the sample. There is no sample deformation between two acquisitions, so the alignment would be more direct, I would say. Then it's somewhat limited to the combination which is proposed. So, I mean, if you need another information, maybe you need to create a new machine then. And that's something which was mostly developed in preclinical imaging, but as I was saying, it's coming now in our field also. I'm from microscopy field myself. The second solution, that will be the one I will be more focusing on, is basically what is widely called correlative microscopy, not only in correlative light electron microscopy, but more generic, where here you move the sample between the different modalities, between the different systems. So you can use that's something also which is used in medical imaging, where, for example, you use ultrasound and MRI, whatever, and then the big key in that case is to register both modalities in order to fuse the information coming from the different modalities. That's also what is used in correlative light electron microscopy workflow, with plenty of flavor of different combination of systems which can be made. And That's really the kind of microscopy, the claim one, which has been pushing correlative microscopies and developing a lot of approaches for that, with some, for example, pioneering work from Wanda Kulczynski and John Briggs and different people at the EMBL as well, on imaging live samples and deciphering some mechanisms of cluster information, for example. So the good thing, it's basically just limited by your imagination about what you can combine together. And the bad thing is that there is a few technical details to be certain. It's not that easy, actually. If I go to the example of CLEM, now it starts to have a lot of, I would say, standard workflows, which has been developed, but due to the work of a lot of people on a big community in CLEM, and there is still a lot of different points to be very careful about, like when you do the fixation from your sample, how you preserve the fluorescence, how you will target the white area, which is also known as a needle in the A problem, how you merge different information, the data set, how also you can deal with the sample transformation or deformation, or maybe finding protocols which preserve the sample from deformation. And there is also a lot of people working in multimodal probes that would be visible in different modalities in order to help this registration. That's basically the method section of a paper. So a lot of people will not care about, but I guess we are here because we do care a lot about this method section. And that's also something that we wanted to push in the Cumulus network. So I'm just going to give you a few words about this network and what it's becoming now. So as you know, the talk today is part of a series that involves this section. So the purpose of this section was to bridge what was done in the life science medical side or preclinical side and the biological side. And we were funded by COST, so the action was led by Andreas Walter. And we got this funding and we organized in different working groups, some of them trying to gather the knowledge about this pioneering correlative microscopy workflows from CLEM, also people working from the preclinical hybrid imaging field, this integrated system as well, and working together also to develop new correlated multimodal imaging pipelines, maybe parsing the different scales also. And of course, this relying also on the involvement of different people from software. This is done now. I mean, the funding just finished in 2022. We were lucky enough to have a following up grant also funded by COST to develop a particular device to help the correlative microscopy workflows, and there is a core team for that. So that will also offer you and any people interested some grants or so if you're interested, you can just go to the website about that. And also, very beginning of this year, we got funded by the Chan Zuckerberg initiative from Act 23 to 25. And that will be the occasion to go on with our activities. Just for you to know, Nubias also, thanks to Robert, has also get funded for the continuation of the activities. So basically, what is going to happen in this Cumulus Globe funding? So first of all, we want to extend to different continents and not only Europe. That's why we have Globe inside it. The purpose is really to create a society to make it sustainable, these activities around multimodal imaging. There will be some industry board setting also. We will be organizing some conferences. Importantly, we want to work a lot with Euro-BioImaging and with people from the synchrotrons as well. From the scientific side, there have been in the first Cumulus different showcase projects that are now getting finalized. So these new novel workflows of the multimodality and different topics, aiming at demonstrating how useful is multimodality. And there will be the occasion to define 3 new ones. So if you're interested to post some, please raise the hand. And we will be organizing also image registration challenge and hackathon. So I've seen in particular Natasha and Joachim are in the room. So they will be also able to answer your question about that. We have been setting up a few repositories to help people finding the good tools or the different workflows. And there will be also some mobility fellowship also in partnership with Euro-BioImaging and the different synchrotrons. You can stay updated by following the website, and there is a mailing list you can register to by using the contact form on the website. So among the different activities, I was saying we have been setting a repository to help people to know what software can be used, for example. And this is based on another repository that was done under the auspice of NUBIAS, the Bioimage Informatics Index. So a lot of people here in the room have contributed. Now, once again, Joachim did a lot for this part also. And here the idea is really to have identified the different key elements of or need in software in a multimodal imaging, so segmentation, registration, visualization, and list different software which can be useful. And for example, in registration, so registration, which is the process of transforming different set of data in one coordinate system, there is these tools which has been like curated by the community as useful for multimodal imaging. And among them, some were coming more from the medical field and are now more and more used in the microscopy field. I don't know if you see my mouse. Can you see my mouse? Okay, good. And myself, I've been developing also one tool which is called ECCLEM. I'm going to tell a little bit more about that right now. So the purpose of ECCLEM was really to offer you different tools inside one plugin to facilitate your correlative microscopies workflows. For people who do not know about registration at all, what you have to know is that there is mainly 2 big approaches. So there is an intensity-based family of registration and what is called landmarks or fiducial-based registration. So intensity-based, I will not talk about it today because the plugins I developed are all based on landmarks and fiducial. And by fiducial, what I mean here is anything that you can automatically extract from your image or manually extract. And that will be merged with the matching fiducial in the other image. So once again, this extraction of fiducial can be automatic based on automatic segmentation, for example, or just count by hands. So there is actually 2 big approaches in ECCLEM. One is the classical ECCLEM, where you do match yourself by pointing matching points on the 2D or 3D images, what should be registered together. And the second one is basically extracting some thing in your image, so that it can be fiducials or it can be surface of nuclei or surface of a cell, whatever. And you extract also the same thing, you segment the same thing on the other one. And then there is a auto finder that will propose a matching and then compute the transformation. So the purpose behind ECCLEM was also to try to keep it simple and proposing tools that has not so many parameters and that people can use kind of easily, which is very flexible regarding the format of images, the dimension of images. So you can do whatever you want, basically. So it provides rigid or non-rigid possibilities. You can do point-based registration or line-based registration, for example. And really, the idea is to help you to construct your own workflow and assisting you in your full correlative microscopy workflows. There's been a new version, which is called ECCLEB 2, and which is actually much more faster, in particular for the 3D computation or the automatic 3D transformation, which is less greedy in memory. And it also provides new tools for the error estimation. I will talk a little bit about that, and also new tools to ease the correlative workflow construction. And I will also describe it a little bit, and some new tools also for how to exploit your transformations that I will also detail a little bit. So the first thing indeed, which can be useful when computing a correlative microscopy workflow is most of the time you have two modalities that maybe you want to really merge at the end, coming from a 3D sample, and the images can be 2D or 3D. And it's very useful to add other modalities or other magnifications, for example. Most of the time that will really help you to find how to merge together the real images you are interested in, because there will be common things in the image that will be easier to find, for example. And what you have to know also is that when you do registration, what is important is that there is a coordinate system which is associated to any element of the image, to the image itself, and also to the objects, the 3D one or the 2D one. And when you compute a transformation, basically you can go from one image to another one. That will give you one transformation. You can compute, for example, this image to this image, that's another transformation, or this image to this image. And because this is just the transformation can be represented as matrix or other kind of representation, but they can be combined mathematically to form one transformation. So it means that if you have computed this pair of image, this pair, for example, and this pair, based on the tools that we provide, you can compute any transformation and go directly from here to here, for example, or from here to here. They are all linked, and you can just go from one image to another one just by computing these three transformations. And this is provided in the cascaded transform, invert transform. That's what we use, for example, in this work with the synchrotron from UK with Maria Archeolaki, and exemplifying it on a study of reovirus. And here, as you can see, the purpose was to merge structured elimination microscopy, so realized in cryo condition, thanks to the work of people from Oxford University, and the soft X-ray images that you get from the beamline at the synchrotron that will give you tomograph in 3D of the images. And for this, as you can see, it's really helpful to have this intermediate image, like the bright field version of the fluorescent image, the low mag X-ray mosaic, and then the tomograph itself. Okay, and then you get a lot of different images from the same sample. This time they are all in the same condition. It was on cryo condition every time, so we are not supposed to get any deformations then. And that's what we did indeed. We did this pair of registration, meaning that we registered, for example, only the bright field and fluorescence image, the bright field with the X-ray, which was kind of easy because they are very similar in appearance. And then the tomograph, the 3D tomograph, and the 2D X-ray mosaics also, because also, once again, they are quite similar. And from these different pair of registrations, then you can construct only one big registration. Here it was only in 2D. And thanks to ECCLEM, you can apply this 2D transformation to a 3D volume. Okay, so it's not aligned in 3D, it's only repositioned in 2D. And that can serve then as an initialization, because now it looks a little bit more easy to register for a proper 3D registration, taking into account the tilt, for example, that you can see it, and the difference in resolution in Z. So for now, the interface is kind of not easy, so that's why it's still in beta version, but quite very soon there would be a nice interface where you say which image you want to register, and that will help you to know what is missing as link and this kind of thing. So that's the kind of results that can be obtained thanks to the workflow. Here on some mitochondria examples, just to validate the techniques, and the other one and the virus by themselves. The nice thing is, because of that, we did not need that much of fiducials, because we have this intermediate image. Okay, so and also what we demonstrate is that there were no deformation in the correlative process, and it was based on some tools that I'm going to describe now. We were able to show that there were no deformation at all during the full workflow. And all the data are available on Zenodo. So what I was saying about showing that it was there was no deformation in this sample, it may happen, and maybe you have seen, I mean, that, okay, even in coming back to the video here, maybe it's easier to see on the mitochondria, but okay, so you have the impression that, okay, maybe the matching is not that perfect. There is, okay, sometimes some small discrepancy. Here it's not too bad, but okay, there is some small discrepancy. This is not perfectly aligned. And basically, that's something you will never reach. So because sometimes your sample deform, it was not the case here. Also because it's super multi-scale, so basically 1 pixel from the light microscopy data here was 100 pixels in the other modality, for example, so that make a lot of difference in resolution. And also because there is no perfect location of any points that you use to register your images. It means that you will get an error, even if you don't have any deformation, even if you are perfectly good, you will always get an error due to the fact that you don't have an infinite resolution and a perfect localization of objects in the microscopy images. So that's something we have developed. So the framework was already partially developed for widget registration, and we developed it for a fine registration as well. And basically what we developed was a method to estimate the error and to get the full distribution of errors for any point on the image without knowing the ground truth. So just using the badly placed fiducials that you have. And just to show that it's working, so here in 2D, but it's also working in 3D on a simple example. So here you have 2 images, and you have some melanosomes in the image, okay, and you have some bits, which are fluorescents in both images. Normally, you will use these bits to do the correlation. Just here to look at the error and everything, I'm going to use these melanosomes, okay, to do the registration, and just look at what's happening in the bits to assess the error. And as you can see, there is some error after the registration, okay? And once again, this is maybe due to some sample deformation, but also maybe due to the fact that I'm very bad in placing my points because melanosomes are kind of big here. And we will check then the error on this, okay? And the usual way to assess how good you are in the correlation process and is basically to try to do your registration using maybe 1 point less. So if you have 7 fiducials as here, you will maybe do 7 registration, but with 6 points every time, and just look at what is the remaining error on these fiducials, because remember, these points, we are pretending that we don't know the ground truth, and it's just to check if we are able to estimate or not the error. So we just know that should be matching. So that's what is usually done. So you do a lot of different correlations, so that leave one out approach, and you look at the remaining error on the points that was not used for the registration to know how good you are or not in the registration, and that then give you a global number, such as here, for example, in 60% of my registration, I was below 0.45 micrometers of error. Okay. And then you can maybe just have a look that it's indeed the range of about the biggest errors that you can get. What we have developed then is basically giving you the full distribution of the error for any points of the image, meaning that for these exact points here, we can give what is the expected error. So then you can represent it as a average accuracy. So for every point, so here, it's more accurate, for example, where are the fiducials, of course, but and it's getting worse on the border of the image, but this is just due once again to the fact that you do not localize perfectly your fiducials, and you have a limited number of fiducials, and you can also give the direction of this error. So here, for example, these ellipses are the area where you have 95% of likelihood to find the matching points. So here's talking about the gravity center of the points which are used. You can see that this one is indeed in the 95% expectation. So this is really the distribution of the error and any point of the image. This has been used in this paper. So for example, and we just checked that the errors that we were getting was actually the one which was predicted by a normal widget sample on a normal widget transformation. Another example here, it's a work with Ori Avinoam from Weizmann Institute on that share. And what they wanted to do was basically prove that lipid droplets could be used as useful natural fiducials for correlation of CRYO-SIM and CRYO-light microscopy. And the good thing with lipid droplets that because they are inside the cells, I mean, as you may have noticed, most of the time the error is less important where the fiducials are. It's exactly less important at the gravity centers of your fiducials. So it's better to be to have them internalized in the cells. And in that case, what we did also was to use sometimes some automatic initialization of the transformation, because and the reason why it was because it was super hard to do it manually, starting from the 3D in very different orientation, because that's the original CRYO-SIM orientation, and that's the image, and as you can see, that's going to be hard to find who is who and everything. So for that, we use EasyClaim Auto Finder, and in this particular experiment, we did automatically segment the lipid droplets in 3D from the light microscopy data. And then for the CRYO-SIM, we just did it manually. We could have worked on an automatic method to extract them, but there were not that many of them, so it was just picked from the image. And from this, you get the initial transformation that was initialized like rigidly, and then based on the error quantification framework I've shown you, we were able to see that it was not rigid, but actually a fine, and a fine actually in one direction only. So maybe that's something due to the resolution in the data. So that's just the kind of imaging data as you get at the beginning in light microscopy and in FIB-SEM. And after the registration, this data becomes this one, and now it's oriented in the right way. So you can go further and look at this affine deformations. They should be square if it was perfectly rigid. And everything, every detail of this process is detailed in a STAR protocol paper as well. So just to go a little bit beyond CLEM, so the tools which are developed can be used also in different flavor of correlative microscopy. We've seen some example of microscopy in order to fuse information and to get the best of different information you can get from different modalities. More and more people are also using it just to validate or better understand a new modality or a new probe that you have. And the basic idea is just to merge an image where you have known markers. So for example, in that context, it was to differentiate different kind of cells and see the nuclei and different markers of different cell types. And in this modality, which is basically a stainless modality based on vibrational optical coherence tomography, a lot of things can be seen, but the question was, okay, what is seen exactly, and what do we see? And for this, we merge and we prepare the correlative workflows with confocal imaging. And these modalities using also intermediate low mag image to do the registration. So everything is in 3D here. And from this, we were able to demonstrate that it was actually only one type of cell, which was very shown in a vibrational optical coherence tomography. And even if you zoom on some of these images, you can even see, for example, that the dense area in the nuclei, which are actually DNA dense also, are not vibrating. So they have no signal and everything. So, and that's then one way to use this correlation workflow, because you compute the transformation between different images, maybe helping you with different intermediate imaging as well. And from that, what we do most of the time is basically, okay, transforming one image in another image. This may have some difficulties like interpolation, which makes that you lose the resolution that you had in your nice image, and in the other way you have a lot of interpolation, but it really depends on what you want to do after that on measure. Another way, if it's just for visualization, to keep the full resolution is whether to transform only the display, superimpose the image using a new coordinate system, and that will be the screen then. And that's allow you to keep the full resolution of the image, still superimposing the image. That's also provided now in the EasyClim V2. And last one, which is not used that much, but I think it's very useful, and I'm going to demonstrate why, is basically to use the transformation that you have computed and apply it directly to the region of interest, because most of the time, you want to maybe measure something or look at something in particular. And the idea is you compute the transformation, and then instead of applying it to the image, you keep your image as they are, and you apply the transformations that you have computed to an ROI, so it goes exactly at the right place on the other image. Then you can do, for example, measurements on the original image without any interpolation or degradation of the image. This is something, for example, we have been using also in different usage of correlative microscopy. So, for example, when you want to to use spatialomics. So on my facility, for example, we had a system to do multiplexing on different proteomic markers, different fluorescent probes, with like 50 different probes to identify different cell population. So you will get the different types, you will get the nuclei and the class for every cell. The thing is that, okay, then you have the cell type, and if you want to go further, like doing some sequencing analysis or any other kind of thing, there is a lot of different approaches, even some integrated system. And we thought that one of them could be maybe to use also a microdissection. And here's the idea of the workflow we have been constructing is to go from this slide to the slide scanner in order to get something that would be very similar to the image that we can get from the microdissector, which is not a quality microscope, but not so good microscope from the image quality. And the idea would be to identify the area of interest on this image and performing different cascaded transform, send this area of interest, so the cells of interest, to the microdissectors to just collect the cells of interest. So, for example, going from the scanner, so here is just a dummy example with some dummy segmentation. We identify a few areas of interest from the slide scanner. Okay, the workflow then we go to the microdissector. We just do another image like a 10x image, even not the one which is used for cutting. You usually cut as a higher dimension, and then you transfer this ROI based on registration on the small image you get from the microdissector, and you send the ROI. And we've been working with MMI as well in order to open their software so we can send back inside the microdissector system this ROI that were pushed. So now we can cut exactly the ROI which comes from the slide scanner, and not drawn manually, whatever. They are automatically identified from this. So, and it's on the whole image. I mean, you don't have to acquire the rest of the image. You just get a small image, and they are here for the whole sample. So this part then is not done yet. So we have identified a few cells, for example, and now we are setting up the workflow to go from the multiplex imaging system to the slide scanner. One last example where it can be used also is that registration and multimodality is something which is gaining attention also in deep learning, because that's a way to leverage the burden of annotations. As you know, you need a lot of examples to train a network, for example. And that's something that you can use, for example, when you do in silico labeling, where you want to create a new modality from, not create a new modality, create images like look as fluorescence data, for example, from non-fluorescent data. So first you need to train with pair of images showing both fluorescent and unstained image. That's also useful when you want to train a network on denoising or image restoration, because you need some example of data without noise that you could then register to the noisy one. And even in instance segmentation, for example, you want to train your network to classify cells or recognize cells, and you need an annotation that could be done by some annotators, but also maybe using some markers that you could get automatically. Lately, we had an example also on the facility, and working together with a PhD student, Cedric, where hematologists from the hospital were asking about developing a system to recognize 30 more kind of cells that they can find in their myelograms. and they do it manually for now. So first idea was, okay, we're going to use deep learning for that, and then we need more data, so we'll use a slide scanning microscope to do so. And that was very nice because indeed we get the full scan very quickly and a nice image. And what you have to know is that usually these people, hematologists, they don't work from the full myelogram. They will choose an area which is actually could be identified also automatically based on the density, but they use a specific reading area to count the cells. So on this reading area, then the purpose was to annotate from the slide scanner the different image. Then they realized that the quality of the slide scanner was not good enough for them. So what we had to do then was to come back to an immersional objective. And as you know, for some of you, there is some slide scanning with immersion objective, but not that much. So our was not of that. So what we did was to use the slide scanner, the bad slide scanner, to identify the region of readings that they will use in order for them to annotate the cells. Okay, but they will not annotate from this. Then we will go to another microscope, a classical confocal microscope, but with a color camera, where we do acquire just a snapshot of any area on the image. Then we do some automatic registration based on, because that's an easy problem because they are kind of similar image. So we just use some classical intensity-based this time registration just to find automatically the area. And then the microscope is derived directly. It's a script that do all the work, and that will acquire exactly, even if the area was here at the beginning, then it will go directly to the right area and acquire exactly the reading area. And then the good thing is that it's directly registered. So the hematologist will annotate his 60 X all immersion image, and still we will have the data from the 40X, and that will allow us, and that's work in progress, to see or not if we can use the 40X for deep learning at the end also. So that's also a question that we had when you want to do correlative microscopy. Okay, you're going multimodal, and sometimes you may want to add this step, or sometimes this step is not existing, so you need to develop a system to add a step linking different modalities. And an example came from colleagues working on mitral valve dystrophy, so a cardiac disease, where they do some quantification of physiology of the valve based on micro-CT data, and also some quantification of the cellularity and the composition of the valve at the cellular level based on the histological slides. And it's not a problem, but it's more like it's not correlative, meaning that you study some of them like this or some of them, and there is not linked between them. And one system was developed in collaboration then with a company which was originally developing a camera for small animal imaging, like infrared imaging. So they modified the system then for us in order, just as it is done already in on some CLEM project, but we image the sample whilst at the same time it is cut, and to make it an imaging modality by itself. So this is a microtome which cuts the slice when you do when you want to do, for example, histological data. So this is also working with CRYO-STAR, which is another way of cutting the different samples. So that's an example of a zebrafish acquired like this. And basically, it does not change that much the histological slide staining process. So you get your organs, you cut it, and then you may want to do your correlative histological staining. The only thing is that in addition, you get this 3D image at at the same time. So you get here are some different examples here from oops, sorry, from an embryon, and I think running whatever. You get some example of images of an embryon, and because you get the slide directly from the image, you have a direct matching that it's easy to do widgetly using ECCLEM also, and then the slice is correlated directly with the 3D dimension. And we use that, for example, here to link the micro-CT to this image obtained from the microtome and the camera in front of the microtome. So you have two 3D volumes, which are kind of easy to register, at least realign, even if there is some deformation in that case. You can even do some quantification directly from the images get from the sample, and then you can get the histological slice directly registered with the 3D volume, and getting the 3D information at the same time as the information that you get, and get the number of nuclei that you had, whatever, in different things. So this is also useful for different kind of quantification at the macro scale, I would say, and it's really for big samples. Okay, and this is quantifiable. I'm going through this. This is some example, yes, of imaging from different samples. So once again, this is without any staining. This is just based on the autofluorescence of the samples. So what I tried to show you, it was that the purpose of registration is really to link multimodal data and you get from the transformation. So the transformation, okay, you can apply it to the image, but you can also link areas and link images, sorry, and ROIs as well. And that can have different application, maybe going from a microscope to another one or target a specific area. The fact that you can estimate the registration is good, because then you can also better know which model should be used, which kind of transformation should be used. And you can also maybe use this information of accuracy to have a margin for error, for example. So if you want to cut something, you can add this error just to be sure to have the area of interest in your cutting. You can see this error also as somewhat some kind of multimodal colocalization, meaning that it's giving you some likelihood of two objects being the same or not. And one last thing also is that because I told you that it was more and more used with this situation for helping deep learning training, for example, there is some way from other domains where you can actually incorporate this error estimation in the deep learning training in order to reduce the number of data and getting better accuracy also in your training. So with that, I will stop and get any questions. There is plenty of people to thank. So some people from the micropixel facility, which I'm leading, people from the CareLab company, people from the multiple myeloma project, my collaborators who has been working on the development of ECCLEM as well, and of about the framework, and people which has been using and helping me developing the different software, and also all my colleagues from the Cumulus Quo. And I didn't put all the names from people from Nucleus, but there is plenty of people, and more people than that, but this is, I would say, the core from Cumulus. Okay, so thanks for that, and maybe I stop sharing my screen if there is any question, or yeah.